Hello students, in this video we learn how do we prepare monoclonal antibodies from particular antigens, why there is a need of myeloma cells in preparation of monoclonal antibodies. Now let's see details about all these questions. When a pathogen, a antigen, enters the host body, its B lymphocytes interact with it. After interacting, the antigen it will differentiate into plasma cells. And these plasma cells can produce antibody against that particular antigen. Here, let me remember you, the B lymphocytes are a type of blood cells, particularly WBC. Now let's see this with a bit detail. When lab animal, say for example, mice is administered with a specific antigen, the mice B cell differentiate into plasma cell and will start producing antibody against it. And these plasma cells can remove and culture in an artificial media in a lab, and we can get the antibody from it. Yes, it's true. It is possible to culture that plasma cells in the artificial media in a lab and they can give antibody to us. But there is a one biggest problem. The lifespan of plasma cell is very short, which may be few days to a few weeks, and soon these plasma cells will die. Therefore, we won't get antibodies for a long time. So, what can we do to overcome from this problem? The problem lies in the lifespan of the plasma cell. Therefore, we can use here a cancer cell known as myeloma cells to overcome the short lifespan of these plasma cells. Myeloma cells are immortal. It means they do not die. How can we use the immortal property of myeloma cells into a plasma cell? The solution is making these two cells hybridoma, means fusion of plasma cell with the myeloma cells. So after making a hybridoma, now they are able to produce antibodies as well as they will have the a property of immortal. Let's look at this with some more details on a hybridoma technology, where mice are first injected with the specific antigen by the intravenous. Mice begin to produce antibodies against that antigen in the plasma cell, and the spleen of the mice is enriched with the plasma cells. Now, the spleen is removed and the plasma cell is separated from it, and it isolated and fused with the myeloma cells. Usually, this fusion of myeloma cell and plasma cell is carried out in a polyethyl glycol, and the fusion is random process, means every time every plasma cell will not fuse with myeloma cell. So this is the another problem here. So as a result of random fusion, here we will get different kinds of cells, that is unfused plasma cells, fused plasma cells, hybridoma cells, unfused myeloma cells, and fused myeloma cells. So it's a task to isolate only hybridoma cells among these all different types of cells. Now let's see how to differentiate hybridoma cells in these different types of cells. One thing is for sure here, we do not need to kill unfused plasma cells and fused plasma cells because they have a short lifespan, so automatically after a few time it will die. So only three types of cells are now left, that is unfused myeloma and fused myeloma and hybridoma cells. Now these remaining cells are taking in a HAT media. HAT means hypoxanthine aminopterin thymidine. Whenever any cells divides in the artificial media, before it divides, it has to make a copy of DNA, and for that it requires the synthesis of nucleotides. Usually this nucleotide synthesized is possible by two ways. one is salvage pathway and another is de novo synthesis. Now in HAT media, cells cannot go through de novo synthesis. This is due to the presence of aminopterin in the media. So now question will be arise, why in presence of aminopterin the de novo synthesis is not possible? The answer for this question is, for the de novo synthesis of nucleotides, it requires tetrahydrofolate which is formed from dihydrofolate, and the formation of tetrahydrofolate can be blocked by the inhibitor aminopterin. Overall, de novo synthesis is not possible, which means only one option left now, that is salvage pathway. But for this also there is one condition, and this condition is this cell must have one enzyme called HGPRT, which means hypoxanthine guanine phosphoribosyl transferase. Remember, the plasma cells have this HGPRT enzyme, but myeloma cells they do not have this HGPRT. Therefore, in HAT media, the fused and unfused myeloma cells die and hybridoma cells alive because it contains the HGPRT enzymes because of its fusion of myeloma and plasma cells. And the fused and unfused plasma cells automatically die because of short lifespan. So separately, there is no need to kill the plasma cells. So finally, we get only hybridoma cells in a HAT media, and it will produce particular monoclonal antibodies for a long time. I hope this video is helpful for you. Thank you for watching this.